this is the second in our Forest Resources Department seminar series for this fall. It's great to have you all here. We have a couple more of these planned. Rebecca is going to be hosting one on November 20th, Interdisciplinary Art and Ecology Residency at the Acreage, a retrospective conversation. Does that sound right? That's going to be really a great one. I hope folks will be able to join us for that. We will be sending around announcements about that. And then either December 9th or 10th, so that'll be either a Monday or Tuesday evening early in December, we've got a contingent coming from Finland. They are social science researchers working in sort of the bioeconomy and forest space. And they are going to be speaking at a seminar as part of this series as well. So we're excited about that. But for today, we've got, well, and the theme of this series is integrating research and extension. And we've got 2/3 of our aquatic invasive species team, extension team here to talk today. They are Dan Larkin, extension specialist, and Megan Weber, an extension educator. I have been hearing about the good work of this team for a number of years, and I'm really glad that they were willing to come and present this seminar. And they're going to be talking about integrating research and extension through the world of aquatic invasive species. We live in the land of 10,000 lakes, many more, and they're doing really important work, and I am glad that they're willing to be here. So Dan, I'm going to hand the floor and the screen to you. You might check this again, because with my monkeying around, you may need to do something to advance the slides.
Okay.
But
Great. Thank you, Eli. Good to see you all. So I'm going to give the first part of this talk and then turn things over to Megan. And I'm going to start by talking about data and new data sources that we've been leveraging in the aquatic plant ecology world, and how that feeds into our research, and then to go along with the theme, how our research ties into extension and vice versa. Really try to make those reciprocal things where both are being informed and improved by the other. And I'm going to talk about the ecology impacts and management of aquatic invasive plants, and end by briefly touching on a couple of our extension programs, and then turn things over to Megan, who will go into greater depth. And I was also going to talk about statewide management of invasive wetland grass, and then I realized there was way too much content to cover, so another day. But this is kind of the premier guide for aquatic plant identification for the upper Midwest, and I just wanted to do a little pandering that the author says, a photographic guide to our underwater forests. So you can just think of these as trees that are underwater and aren't woody. But kidding aside, it's, I think, an underappreciated source of biodiversity in our region and in temperate ecosystems in general. These plants perform important ecosystem functions and services, but they're hidden and they're difficult to sample, which comes up in sort of good and bad ways, interesting ways. So you'll often hear the term macrophytes, which just encompasses aquatic plants as well as macroalgae. And that spans this moisture gradient from emergent plants that are growing in moist soil all the way to stuff that's in deeper water, free floating and submerged. And I mentioned that they're hidden and difficult to sample, and there's sort of a silver lining of that difficulty in sampling. So I also work in terrestrial plant ecology, and any ecologist who's going to go out and measure a prairie might do it in different ways for their vegetation sampling. And so it can be hard to integrate data from across monitoring efforts. But because aquatic plants are difficult to sample, there's actually been widespread adoption of a standardized method. So this technical note was published in 1999, and it proposed a standardized grid-based point intercept survey for aquatic plants. And in this region, at least in Minnesota and Wisconsin, the agencies and consultants and everybody got on board sort of miraculously. And so now we're approaching 25 years of these data that can be aggregated. And what this looks like in practice is that somebody is on a boat and they navigate to a predefined point using their GPS, and then they use a double-sided metal garden rake, either dragging it on the bottom of the lake on a rope or twisting it on a pole. They pull up the sample and then they identify each plant to species and they give a rating of its abundance. So you get sort of a measure of relative abundance. And this is what these data look like. So this is from each of these X's is a point that the surveyor would have sampled. This is on a shallow lake, so the whole thing is littoral or vegetated zone of a lake. Obviously, many of our lakes, it would just be kind of like a shoreline fringe where you'd have plants. But they go out and sample each of these points, and then in this case, we're showing where one species is present with these yellow circles, but you know, you get this distribution information for every species that they find. So these data can be synthesized really nicely, and also we get data at multiple spatial scales. So we have sampling at the point scale, which I think of as kind of the plant neighborhood, like where a plant is living and interacting with other species. But then that information gets aggregated up to the lake scale and then across lakes to regional scales. And what's also nice is where there's been repeat sampling, we can see change over time. So this particular survey is showing a lake where there was a lot of an invasive plant called curly-leaf pondweed. You can see it essentially covered the entire lake. They did treatment. The treatment was highly effective, and then there's just sort of scattered little spots of it after that. So we can look at change over time. And so I'm going to highlight a few synthesis efforts from the last few years. The first was by Mike Verhoeven, a Ph.D. student in conservation sciences, who worked with me. And Mike threw himself at this and compiled about 19 years of data from the DNR, from local units of government, from private consultants. His data set ended up including over 1,500 lakes, over 3,000 surveys, over 370,000 individual points that were sampled. And what's exciting as an ecologist is it's over 500,000 species observations. And then there's often environmental data associated with these lakes or this sampling that we can further use to look at what's driving community patterns. So that was just for Minnesota. And then Heidi Rantala from the Minnesota DNR and Zach Feiner from UW-Madison got funding from Midwest Glacial Lakes Partnership to bring Minnesota and Wisconsin aquatic plant and fisheries data together to look at relationships between aquatic plants and fish. So that's an ongoing project that's essentially, you know, doubled the access we have to these types of data. And then finally, Alex Badges, who's a quantitative ecologist with MACERC, which is a Minnesota Aquatic Invasive Species Research Center, created this awesome website called PI Charter that's a sort of a living data portal. Those other examples were just snapshots in time, what's, you know, all the data we can gather. But Alex has created PI Charter, which has a largely automated upload, so people can contribute new data, and it has these really user-friendly interfaces for data exploration. So all of this has just really created these phenomenal data sets where we can try to answer questions about aquatic plant communities that wouldn't have been possible prior to these efforts. And of course, none of this would be possible without the blood, sweat, and tears of countless people who performed these really time-consuming surveys. And so this is Alex's sort of leaderboard for Minnesota of the groups that have contributed these data, and it spans many different organizations. And then when you add on Wisconsin, it's many, many more. So I'm going to turn now to invasive aquatic plants, which is what Megan and I work on for our AIS extension program. This is data from the Minnesota DNR. They have an infested waters list where they will designate a lake as infested based on the presence of certain species. Here I just subset the plants that they use to designate waters as infested, and you can see the increase over time. And this is on a log scale, so these are pretty big climbs. And then there's many more that are not even tracked. They're sort of off the scale because they're so widespread and common, and that includes curly-leaf pondweed, which I mentioned before. It's been here for over a century, so it's really widespread, probably in close to 1,000 lakes. And then some emergent invasive plants that also don't get tracked. Okay, so why do we care about these plants? There are a number of reasons to. You know, largely in this state, it's driven by human use. It interferes with recreation. They have economic impacts like reductions to lakeshore property values. It can reduce habitat quality for fish and wildlife. And then in my group, we focus a lot on threats to native plant diversity. So I'm going to spend a little time talking about the ecological consequences of these invasive plants. First off, looking at sort of statewide trends in invasions and native plant diversity, and this was leveraging data from the Minnesota DNR Shallow Lakes program. And you can see on this map, the monitoring came from all of these lakes, some of which were invaded in black or uninvaded in red. Some of them we just have a single survey. Others that are triangles, we have a time series. So it's over 1,000 lakes, 13 years of data. And these next few slides are going to be from work that Ranjan Muthukrishnan did when he was a postdoc with me, and he's just started as an assistant professor at St. Olaf. Yeah, you know Ranjan? Yeah, he did. Yeah. So you'll see him. I've already seen him at talks on campus. Yeah, and he's living, I think, in Minneapolis. So, yeah, great. Yeah. No, that's great. Okay, so the first thing we looked at is diversity invasibility patterns. That's something that's been looked at a lot in terrestrial systems. And we found that the most diverse lakes were most vulnerable to invasion. So here we have lake level species richness on the x-axis, invasive species richness on the y-axis. You can see there's a positive relationship. This is called the rich get richer, and it's a commonly identified pattern where the most diverse areas accumulate the most invasive species. And then so this is at kind of a regional scale, and then what you hope at a local scale, what's often seen in small scale and experimental studies is what's called biotic resistance, where higher native diversity confers resistance to invasion. Using the time series data, we did not see that pattern. So if we had seen it, it would have looked like this, where when you had higher native species richness, a point was less likely to become invaded over time. We didn't see that pattern, but we did find competitive exclusion, which is another mechanism where invasion leads to a reduction in native diversity. So it's sort of it's an unfortunate sort of double whammy for these native species. Now, we are now revisiting this apparent lack of biotic resistance. So Abba Panda, who's a third-year PhD student, is revisiting this diversity invasibility relationships. She's incorporating more data and more environmental context. And I think what's really important is looking at relative abundances of species, not just presence absence, which can be a less sensitive measure of these interactions. So more to come on that, and we suspect that there may be aspects of species density and traits that could be used to sort of build in more resistance to invasion, and ultimately we hope to extend that to revegetation and restoration of lakes. We're going to talk about another type of diversity change, which is biotic homogenization. And handily, I already used a forest example for this. So this is a cartoon landscape where we have 4 forests, Each of these forests has 4 species of trees, indicated by different colors. So there's a species richness of 4, that's the alpha diversity, the local scale diversity. And then also the forests are compositionally distinct from each other, and that's called beta diversity. So they have some species in common, but they don't entirely share species, so that there's beta diversity or turnover in species composition from site to site. And that's, you know, one of the qualities we like from a conservation perspective is sort of the uniqueness of these assemblages. In contrast, landscape 2, if we just looked at richness, we'd be just as happy because there's still 4 species per forest, but the forests aren't distinct. So we've had a loss of beta diversity. So we looked at this in aquatic plants, again, using the same data set, and found that lake plant communities were undergoing biotic homogenization. And on this figure, I'm going to show you the results for uninvaded lakes as well as invaded lakes, and we're measuring change over time. So there's an initial measure and then a final measure. And when we have more species in common or lower beta diversity, we'll have higher values. So first off, with the uninvaded lakes, we see that the lakes were becoming more similar to each other over time. This is in that 13-year period. And then that phenomenon was magnified in invaded lakes. So we are sort of seeing this erosion of the uniqueness of the lakes. And this is not just a result of the invasive species themselves being there. If you sort of subtract out the influence of the invasive species on composition, we're still seeing this erosion of distinctiveness in native species. Now I'm going to talk about a couple of these species specifically that receive a lot of management attention. And that's Eurasian water milfoil and curly-leaf pondweed, which I've already mentioned. And one of the features of curly-leaf pondweed that stands out is that it grows early and tolerates these low light conditions. So this is going to be going back to Mike Verhoeven's dissertation. And what Mike wanted to do is look at the ecological niches occupied by these species with the idea that while they sort of get equal attention as common invasive plants in lakes, that they may differ in their capacity to competitively displace native species. So are they likely to be causing direct competitive displacement of native species or perhaps occupying new niches? So I'm going to show you results for curly-leaf pondweed, will be in black, Eurasian water milfoil in red, and then 32 common native plant species are in gray. And Mike characterized 3 dimensions of their environment. Light, which is a big deal for aquatic plants. Of course, it is for plants in general, but there's really rapid decay of light availability in water, water depth, and growing degree days, a phenological measure. And what Mike found was that curly-leaf pondweed grew in areas that were darker, deeper, and earlier. So here, in terms of light, we see this spike in occurrence of curly-leaf pondweed under low light conditions. We see the curly-leaf pondweed shifted into deeper water depths, and we see this phenology we expected of much earlier growth. And so what that suggests is that curly-leaf pondweed is sort of maybe occupying an underutilized niche and succeeding by taking advantage of this habitat availability, whereas Eurasian water milfoil has greater niche overlap with natives and is sort of competing with them where they live. And in a manuscript in prep, Mike found from additional observational as well as in-lake experiments confirmed these niche-based predictions that Eurasian water milfoil is a bigger competitive threat than curly-leaf pondweed. Okay, one more species, which is starry stonewort. This is a new invader to Minnesota. It was found here for the first time in summer 2015, right before I started my position. And so I ended up doing research on it right away. It's a cryptic invader. It's not a vascular plant. It's in this group of macroalgae called charophytes that includes a lot of cool native species as well. And its name, starry stonewort, and its mode of spread is from these star-shaped asexual reproductive structures called baubles. And so this is where I'm going to transition a little bit into management as I wrap up here. There's been a lot of effort to treat starry stonewort as it's found in new lakes. And so we've been evaluating management methods and effectiveness, and we found that chemical control of established populations of this species is really unreliable. So this is showing before and after treatment, and treatment is overwhelmingly with copper-based algicides, as well as some herbicides. And the mean effect is a small but significant decrease in starry stonewort, but that obscures a lot of variation in outcomes, where sometimes you see the desired decrease, but oftentimes there's no change or it can even increase following treatment. In contrast, when starry stonewort is found early, hand pulling of new small populations can be highly effective. And this is something that we work on a lot in our AIS extension programs, is increasing capacity for surveillance. So this is a population that was discovered through one of our citizen science surveillance programs. And they started doing hand pull treatments, and it's really contained the population, kept it from spreading beyond this small area where it was first found. And then we've seen that again in additional Minnesota lakes, sometimes in combination with algicide treatments. This is in really marked contrast to curly leaf pondweed, the species that I showed you before that point intercept survey map, where it had been covered the entire lake and then it was nearly eliminated following treatment. And so outcomes here vary depending on an AIS-related issue and reporting. And then through AIS detectors, we were encountering a lot of management questions, which weren't the focus of that program. So we spun off a new program called AIS Management 101. And this reflects, I think, what is a pretty strange natural resource management scenario, which is that management of these species is highly decentralized. It's not like the DNR is deciding we're going to control in this lake. It's really driven by people who live on a lake, whether they have a lake association, how organized and funded that lake association is. So there are all these people out on the landscape who don't have resource management or lake-based professional experience who are making these management decisions. And so where my mind always goes, I don't know anything about cars. You know, I would take my car to Jiffy Lube for an oil change, and then they would say, oh, you need these fluids, you need these filters. And I would just be sort of at their mercy, like, do I believe them? Is it worth the money? And that's where these folks are, but in a much higher stakes situation of whether or not to treat lakes with chemicals, which chemicals to use, is it worth the money? So we developed this program called AIS Management 101 to sort of give people, non-professionals, a foundational understanding of issues related to plant management, pesticides, how management effectiveness is evaluated, et cetera. And now I'm going to turn it over to Megan.
Okay. Thanks. Okay, so I'm going to take in now all that Dan has talked about and bring in some other of our extension programs and how research feeds into those, and likewise how our extension programs feed into research. And I started kind of with things that are under the most development, and it will go down to our more long-standing established programs. So the first one that I want to talk about is one called Zebra Mussel Safari. And this came about really as a response to community questions and concerns. So in lakes that have existing populations of zebra mussels, people want to understand what's going on. Why does the population fluctuate? Like this year was really bad, and now this year I hardly see any. Why is that? And in many cases, we don't have the data to give real specific answers to that question. There's also a lot of questions around management. Management of zebra mussels in Minnesota is still very new. Oftentimes, permits are granted only on an experimental basis. There's a real limited number of products that are available to do control. And so that means that before permits are approved, having more data about what's going on in that lake environment, what the current distribution is really important to helping to get those approvals. So there's these two pieces that people came and said, like, we need help. We really want, and we want to be able, we have mussels. We've had these plates that are used to sample for zebra mussels out in our lake for early detection. But like, now what? Should I still be collecting data? And the answer is that, like, yeah, there's some really useful data in there. But we have to figure out how to put that together into something that's usable to answer research questions, in particular, some of the ones that the community are coming together with. So that's where we've partnered up. This has been a pilot program now since 2021. So we're in this was the fourth summer of pilot phase for this. But we have community members who live on the lakes who act as liaisons, in particular to other homeowners around the lake, to help have these samplers around and collect data that can come in through this program. And we've learned a lot. First, like the real basis here is even how do we collect this data? And part of the reason that this is a big challenge is because plates end up looking like this. The plates go in and out each year. So you're getting new settlement. This is really looking at what reproductive pressure is. So you're getting tiny mussels. You don't have like long mussels that are a year or two old that are really easy to see. You have plates that look like this. Almost all of these are zebra mussels, not all. There's probably a couple of things on here that aren't. But it's meant that we need to figure out new tools that can be used for people to count this, because, you know, with plates that only have a few, like, that's pretty easy. But even then, like these, like I said, these are real tiny. So some, you know, like this is a mussel. This is a 6-inch plate. So maybe for reference, 6 inches from here to here. And, you know, people have varying ability to spy these. In particular, if you're not very familiar with them, it might be harder to realize that, like, that one's a mussel, but that one's a snail. And, you know, depending on how great your eyesight is too, like, you can get a lot of variability in the data. So we had to figure out a way for that to happen. And one idea was this photographic method. So in this earlier slide, you can see this is one of our volunteers, and she's taking photos. And the hope was that maybe we could just use these photos to do that work. It got to a point where MASERC was ready to sponsor a grad student to do this. And now this is feeding into graduate student research. So that's Sawyer. I don't know, Sawyer might be online too. I'm not sure if she ended up joining. But Sawyer's taken this on now and has found this really great tool to help use to count plates. So here's now, it's counted this plate for us. So there were 2,528 zebra mussels on that surface. So you can imagine what the time input would be to do that. And then you think about each plate in that stack I showed you. So there are 3 plates. Each plate has 2 sides. And then there's 3 plates. And then each lake may have, you know, the lake that I showed up there had, I think, 26, 28 plates. So you can imagine the amount of time input that's required. So it really requires better methods in order to have bigger data sets to do these analyses that we need answers to. So this is a tool that Sawyer found. It's called Count Things. Believe it or not, it counts things. And they helped and developed a package specifically for zebra mussels that helped with this situation, so that we have now this tool that we can start to integrate into this type of a project. So Sawyer's been doing a lot of really great work to get this set up into something that then can become a longer standing extension program. Yeah.
So is there also a differentiate things? So it didn't just
count
every item.
Correct. Yeah. So the question is, can it differentiate? And the answer is yes, it can. So it is counting mussels. And when I'm done, I snuck in after the thank you slide some other pictures that'll help show that. So remind me to come back to that at Q&A, and I'll show more. But yes, so it's only finding the mussels. So it's figured out how to tell which of those darkish colored shapes on this plate are a mussel and which ones are not. Okay, and part of Sawyer's research project, I should say too, she went out, she has done hand counts of the plates. She put photos that were taken by volunteers through this Count Things. And then she got this fancy light box and took these really high quality images and ran those photos through Count Things. And then also did like a Count Things plus, I'm gonna go in and manually edit their counts. And there's, in general, like this does really well without needing, like, you can go in and adjust if you wanted to, but if, you know, you're getting hundreds of plates in, you're not maybe gonna spend time here. And so the difference wasn't huge. Like I would be happy with the data that came in without having to even go in and manually adjust. Okay, so going on to our next invertebrate is, this is a spiny water flea. So again, another tiny invader. This is one on a fingertip. And it's a predatory zooplankton species, first detected in Minnesota in 1990. And this is a spot where research that's been happening has identified needs for additional surveillance. And that's a spot that Extension has been able to help kind of come in and fill those roles. So this is Don Brandstratter, and some of the work that he did found that there's this significant lag between the first detection of spiny water flea and when it was actually introduced to the lake. And he did that by taking sediment cores. So here you can see this green line. So that's the first reported detection. And then these two arrows are where the sediment cores say, no, we actually found them this far back. So this is almost a decade difference from how long they had actually been present to when they were first detected. And this is a species that lots of our other detection methods aren't likely to find this species. Like throwing the plant rake isn't likely to find it. They're not going to settle on those zebra mussel samplers. So the things that lots of people are using already to do early detections aren't going to be the best tool for detecting this particular species. So through this, they've said, okay, so like we have a need to like do maybe more focused surveillance for this species because we're not getting a lot of new detections also. So since 2016, I think there've only been 2 or 3 other new detections in Minnesota. And certainly there's more lakes that are suitable for this species to be present in. So Don, Val Brady, and Meg Dewar, who was the research outreach specialist at MASERC, got the base work done for this program to find a way to create something that volunteers could use to help build into surveillance. And that's where spiny surveillance came from. And now this program has come in to Extension to be a longer lived program within our volunteer life cycle. So we have volunteers now. We do a training, they get a webinar, they go out. This is Kevin, a volunteer who does a number of our different programs, and he's monitoring here in the Twin Cities metro area, which is also a space that hasn't typically been monitored. I'll get to that in the next slide. But this is just last year's data. So last year, that was 70 additional surveillance trips that probably would not have happened otherwise, 29 different lakes that are getting monitored, and no new detections. So that's always good news too. But this is also how this is getting this reach beyond. So this area circled in yellow now is kind of where spiny water flea are typically found in Minnesota, with the exception of Lake Mille Lacs. So right here also has spiny water flea. But in general, it's this northeastern part of the state that has it. And that's where a lot of attention has been paid. But there's certainly concern outside of that region. And by being able to tap into this larger volunteer network, we're getting people who are monitoring in the metro area. We're getting this kind of like north central lakes region. So there's additional monitoring that's happening now that's all been identified from these research needs. Oh, here, there's arrows. Okay. Then this is the longest established program. So this is Starry Trek. And this is a program that's really focusing on starry stonewort. So Dan talked about this species already. And one of the things he said was early detection is key. So that's one of the things that this program is seeking to address. So this is a statewide effort. The little markers here are where we had sites across the state just this year. It happens once a year, and we partner with local organizations. So each of these marks is like a soil and water conservation district, a county, a local lakes group that offer to host a local training site so that people all over the state can go out and do monitoring all at the same time. And ultimately, this has increased the statewide capacity for monitoring for starry stonewort. They're also looking for other species too, but we really want to focus on starry stonewort. It was originally born after it was found in 2015. There was a flurry of new discoveries in 2016, and we had just launched that AIS detectors program that Dan mentioned. So we had this new network of volunteers and said, like, gee, this is something that it seems like that group could maybe get involved with and be helpful with. So we had started this program in partnership with Wisconsin, was also hoping to add in a similar event. So together we created Lakes Protocols, and we actually have sister events that happen on the same day each year now. But last year, this is what happens. We had 194 people who came out across the state. They were able to look at 252 water bodies And then on those water bodies, ultimately search 297 public water accesses all in a morning. So this is done by noon on a Saturday. So again, it's this way to add this capacity to aquatic invasive species efforts in the state. And part of what we do involves incorporating research into that. So here's some more work that Ranjan did when he was a postdoc in Dan's lab. And he was looking at what conditions lead to like higher risk in a lake. So which of these lakes are more likely to support a starry stonewort population? And then we can use this data to help direct people to these higher risk lakes. So within this program, we create the monitoring routes for each of those dots. We send them out the routes. And this is one of the ways that we're selecting lakes to send people to, is by looking at what are the higher risk lakes. So ultimately, we've had 4 new detections of starry stonewort because of this program. So volunteers that have been out. So 2 up in kind of the northern part of the state, and then 2 in the central part of the state. And that's amounted to about 20% of the new detections of the species since we launched this program. And then I also mentioned that there's other species that people find when they're out. So they, of course, many people are familiar with other invasive species already. Local coordinators might have something of interest and teach a group about some others. So since we launched this program in 2017, there have been 87 new reports of aquatic invasive species from this program across the whole list of taxa here. And one other one that is of particular interest that also has spurred kind of new research and shown how extension programs can feed back into research is this one here. So this is a Curbicula freshwater golden clam. So this species has been in Minnesota since like the 1980s, but it's typically been limited to river systems, and more specifically river systems where there's some other kind of thermal input to that water body. So like a power plant that has cooling water discharge, or a wastewater treatment plant that creates this artificially warm habitat throughout the whole year. It had not been found previously in an inland lake that doesn't have any kind of outside warm water input. So volunteers in 2020 on one of their rake tosses tangled up in all the plants found these 2 right here and reported those in. So this was the first time that we had detected this species alive in an inland lake in Minnesota. So that had some research questions. We went out and there were actually many more. With a couple of more rake tosses, easily found like 30 live clams. I partnered with the local soil and water conservation district to just figure out what's going on over top of winter. Are they surviving? What's going on? And ultimately this is what we had. So this is a frequency histogram based on the size of the shell. And to start, so winter is when we started. We was kind of backwards because you're going to look at the bottom first. There, you can see this shift as they grow. So this is the winter monitoring, and then after the winter, the following summer, at the end of the summer, this is what they look like. So you can see they'll shift over, and each of these little cohorts of clams, you can see have moved up, which means that there's a new cohort here, right? So this one is a new group of clams. So we can see that reproduction likely happened in this lake during that period. And then based on this information and this project and this new discovery, that elevated the priority of the species for research. So now this is one of the list of invertebrates that's a priority species for MASIC research projects. Okay, that's all I had. Do you want to come back up, Dan, with questions? Or do you want me to show? Yeah, sorry. Thanks, Eli. Okay, I'll answer Eli's question first while I'm thinking of it. So here's another, maybe the next one's actually better. Okay, so here's another one of the zebra mussel slides. So back to like, how can this count things that aren't zebra mussels? So I'll let you take a look at it quick. I'm gonna switch it to show you what it's counting, and then I'll talk about it. So there's the point, so that's what it's counting. And then if we look, so lots of this is like, these are plant mat, this is some kind of plant matter. This looks like it's maybe a caddisfly tube. This one looks like it's probably a snail. So there's lots of other stuff that ends up on these plates besides zebra mussels. And it's able to look at, like size, shape. I'm assuming the way like the color reflects in times to kind of discern which things it should be counting and which things it shouldn't. And it does a pretty good job.
not different.
They're not. So originally we were working with someone else to build a system. And I learned all kinds of things about how that works. I'll see if I can repeat it in a way that makes sense because it probably went over my head. But what happens is like as it's doing some analysis, it can put like filters on and different surfaces or different things react differently to those filters. And then it can say, okay, so if it's a zebra mussel, if you put this filter on, it looks like this, and the pixels react in this way. And if it's not, then it may not. So that's, I'm assuming, how this program works as well. In the end, like you don't see any of those filters or anything. What you see is you see this, and then you see this.
Your license expired today.
Oh. Yeah. I'm not, but uh-huh. Okay. Yeah. So I have not heard of it with a cinder block, but my guess would be that it would be able to handle it just fine. I would assume that like you would need to just take photos of additional surfaces. Yeah. But if you, I think the website is just countthings.com. It's fairly easy to find. They count many things. Like lots of the things that I saw on the website were like, like in forestry, or in like forest harvest, like you have a truck and you have a whole bunch of logs and you take a picture of the back and it just tells you, okay, here's how many logs you have. Or like in machining, I have a stack of like whatever number of pipes and I can count that. I think Sawyer heard about it from another student who was using it to count fish eggs. So they have a whole bunch of things that are already built in. I think they initially tried one of the pre-programmed things they had and it didn't work so well. And she had just messaged with them and they're like, oh, well, we'll just build something that works for you. So I think the trick is like, if you message them, they'll likely help you. And then you have to buy a license in order to use it. So yeah. Depending on what, if it's not one of their existing things that they count, then yeah, I think so. Mhm.
Dan, you mentioned at one point that uninvaded lakes were getting more similar over time. I think you mentioned a 13-year sampling period. So lakes that the increasing similarity wasn't due to invasion. What was that due to? What's going on there?
Yeah, that's a great question. I don't know the answer to that. It's other studies of biotic homogenization, for example, in Wisconsin forests have found similar overarching trends. Ours was on a pretty fast time scale. Some of those homogenization studies are over like a 50-year period. So it's potentially sort of habitat degradation due to eutrophication, climate change, other factors, but fundamentally we don't know. Okay.
Given that you have time series, can you look at climate signals in it, or can you look at things like the role of water temperature variation, or I saw the growing degree days, like does that vary from year to year, and those, those types of data to look for model climate change projection?
Yeah, Rebecca was asking about whether we can use the time series data to look at climate change effects, and we have done that. So I mentioned at the end when I was talking about management of invasive plants, talked about curly leaf pondweed management. We found, for example, that snow cover over ice was a strong predictor of curly-leaf pondweed abundance that next growing season, with more snow cover suppressing the abundance of curly-leaf pondweed. So with a winter like we had this past year, where there was very little ice cover, very little snow, that preceded a gangbusters year for curly-leaf pondweed. So with the direction of climate change, we will likely see curly-leaf pondweed benefiting. And yeah, so growing degree days have proven to be a pretty good predictor for species abundances. And I think, the challenge is, getting into that 20, 25-year period. It doesn't give us a huge amount of variation to work with, but there is quite a bit of interannual variability. So I think we can have those correlations, these species do well following these weather, these winter conditions. It's harder to then attribute to directional climate change, but yeah, I think we're good in there.
Degree days that has a signal that relates to, say, abundances or something like that, then could think about projecting what are the projections for that future
Yes.
to project the impact.
Yes, absolutely. And we've, on other work with wild rice, with monitoring data from tribes and intertribal treaty organizations, we've done exactly that. We had sort of 20 years of data. And in that case, winter severity is really positive for wild rice, and warmer, milder winters are bad. And so we were able to show there are strong correlations with that interannual variability. And then if you look at kind of the overall trajectory with climate change, we can make projections. Yeah. Well, so light is real, like the primary driver of everything. So yeah, so temperature interacts with different species in different ways. So I think a more severe winter hurts curly-leaf pondweed because it has that early phenology. And so if there's not snow cover, then it's getting light and it's able to jumpstart its growth. If there is snow cover, it holds it back phenologically. And then with wild rice, it probably has more to do with seed biology and germination, where it wants that long, cold, dark winter to know that it's time to grow. But yeah, light is the biggest factor in lake plant diversity is light availability. And if you have elevated nutrients and eutrophic conditions, that light decays so quickly and you end up with just a few species. Well, you can have a flip away from plants at all to just algae, or just kind of these really tolerant species that can handle that low light.
Question. One of the things that we hear about participatory science approaches is that, well, how reliable or how accurate are the data? So in a situation where the primary purpose is early detection, I'm guessing if someone finds a new lake that's infested, that's quickly confirmed by a professional crew. So maybe that's not too big a concern. But maybe in some of your other data sets, it is more of a concern. So how great a concern is reliability of data collected by volunteers, and how has that affected, or what methods have you developed to deal with that?
Sure.
So I think for the ones, so I guess first I'll say there is a body of research comparing, especially in like invasive species, early detections and such, about reliability of citizen monitoring versus professional monitoring. And it's really pretty good. A lot of it comes down to the training that you give. I like to think we give good trainings. So I think that helps, is, like what level of input are you putting on to teach? But for every one of these programs too, there is like a QC point, or a QA QC. So with Starry Trek, the volunteers are going out. If they think they find something, they're bringing it back to their local coordinator. That local coordinator is doing a pass through and will sort out things that they're confident are not invasive species. And then something that they still think needs a professional eye then goes to DNR for verification. So that all happens before those points are released and any kind of new infested water is declared. For like same spiny surveillance has that same process where like they have a way to report in what they're finding and that's getting reviewed. We have lots of people who submit pictures of all kinds of various zooplankton. And so far they've all been some other kind of zooplankton that we're not concerned about. And then like with this program, the one that was up was Mussel Safari, same thing. Like they're taking the photos, but they're not actually counting the data or deciding which thing is a zebra mussel and which one isn't. So I think, that the design, both in terms of training and, what your QA QC is before release are the pieces to think about.
Yeah, and also picking the target. So Megan and I developed a program called AIS Trackers, where citizen scientists were going to go and do lake monitoring and collect vegetation data and evaluate the effectiveness of management. And we did a pilot program, like came up with a curriculum, did a pilot program, and then realized this is just not going to work. Like it's overwhelming for people. It's too challenging. We're not sure that we can trust the data. And so that's what led to AIS Management 101, where our participants in our pilot program were like, we don't want to collect these. We want to learn more about this stuff. We want to understand things better, but we don't want to collect the data. So we pivoted. Yeah.
Are there extensive databases covering riverine lakes or like tidal lakes?
Yes. Yes, they do show up in there. So for example, if you wanted to explore PI, if you just Google PI charter, you'll get to the website and you could explore that. And if there are particular riverine lakes you're interested in, they may show up. Yeah.
Have you seen change around people who will become what's happening?
That's a good question. I don't know that I've personally seen change yet, at least. Yeah, I don't know. What do you have?
Yeah, I don't have the Minnesota time horizon for that. Like I know that the houses have really changed, right? From like the little ramshackle, the size of them,
Quantity.
quantity, and fanciness. But I
That's changed too. So like there has been a trend towards more development. So undeveloped shoreline, there are more homes on lakes than there had been previously. So like there is more density too.
and the demographics of our programs lean on the older side, as you'd expect. People who have a lake home, are retired, have time. But we've gotten more young people. I think we're sort of bimodal.
Yeah.
Now, where we have a lot of retirees and then a lot of early career people, young professionals that are doing internships for the DNR or something.
And I'd also say, I don't think we have a single program where you have to live on a lake to participate. Every single thing here, the Starry Trek, that's all done at public water accesses from shore. You don't need a boat. You stand on shore and you toss a rake. And that's where boats are coming in and out are typically some of the places that we have the most first detection. So it's a reasonable place to send people. So standing from shore, standing at the end of a dock, it's a good place to do early detection. You don't need a boat. All you have to do is be able to get to the public access. And then we provide the rakes for that too. So they don't even need equipment. They just have to show up and wanna go. Spiny surveillance is probably the one that requires like the most specialized. Like you do need a boat for it. I know Meg, when she was doing some of the development, tried it out in a canoe, and she was generally able to do it in a canoe. Probably not doable in a kayak without tipping over. But so like that's probably the one that would have like the biggest hurdle in that like you need to have a boat. But everything else that we have, well, I guess sorry, Zebra Mussel Safari does. That one would be the one where like if you live on the lake, then you're the one who has a sampler out. But everything else that we do, there's something that you can do whether or not you live on a lake.
it's been very interesting for me moving here and starting to work on invasive species in lakes. You know, I'd worked in wetlands and prairies and woodlands in the past on invasive species, and the only people who cared about those things were sort of nature-oriented steward types, birders, et cetera. And so it's a really interesting social context working on aquatic invasive species because so much of the passion and intensity is driven by people's sense of ownership. You know, having a lakeshore property, can their grandkids water ski, what's happening to their property values. And so the level of intensity of feeling and emotion around invasive species in lakes are like nothing I'd ever encountered before.
Yeah, we started, like, I think we noticed this, and in that core course that Dan mentioned, was it 2 years ago now that we changed to have the Slido? We had this interactive activity at the end, and I would love to ask that question. So at the end, we ask the question. It's where it generates a word cloud, and it says, you know, like what are your feelings about the future of aquatic invasive species in Minnesota? Or something along those lines. And then, you know, they put in 1 or 2 words and generate the word cloud. And at the end of our class, the biggest word in the center is always hopeful. There's a mix of like emotions that are around that, as you would expect, but it does say hopeful. And I wish that we could maybe capture that before they took it too, because I do think there's a lot of anxiety and worry. I think we've seen that qualitatively in like our evaluations and the comments. People mention like how they felt before. And so, yeah, I think like the emotions definitely run high on it.
And the messaging around invasive species can be so extreme around aquatic invasive species, where people talk about like it's the death of a lake, you know, your lake has cancer. It's just the rhetoric is so extreme that I think it's sort of backfired.
Mm-hmm.
Yeah.
I ran into that doing work on a terrestrial invasive species where we were like, people were getting mad at us because we weren't eradicating it. We were like studying its biology.
Yeah.
And we didn't want to join because like we're contributing to this horrible, horrible thing that is like, you know,
Yeah.
it's like ruining, yeah.
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 So it's a
the ways people put together information.
Mm-hmm.
But I think the messaging around invasive species has contributed to problematic public outcomes and perception, right?
Yep. And it was, yeah, there was this desire to get people to care, but it led to this militaristic, the aliens are coming and it's a disaster. And so now it's this tricky balance of like, yes, these species cause problems, there are impacts, but it's not the end of the world. Like,
So I have a question that I might know part of the answer to, but I'd like to hear your thoughts on it. So the university conducts research. There are soil and water conservation districts all over the state. There's Bowser, there's lake associations on lots of lakes. There are lots and lots of people out there working on lakes or volunteering or caring a lot and thinking about lakes. What's the role of extension? Why do we need extension as part of, you know, the land grant system? Why not just have Dan give a talk to a lake association? And, you know, do we need an extension educator? Or and the flip side of that is, do you have the sense that if you had 5 Megans, you could 5 extension educators doing this work? Are there 1,000? You know, are there tons of? So what's the role of extension, and how does that move your work forward compared with it didn't exist?
there's only one Megan. I think the key, I was totally new to extension when I started the job here. So I guess now I'm like 9 years into it. So it was new to me and kind of took some time to figure out what it is and, you know, because it can be so varied from program to program what good extension looks like. But I think what I took away and have tried to practice is that extension should be tightly connected to research, and that, as in our title, like we're aiming for this integration of research and extension. We don't always get there, but that's what we strive for. And so there's a lot of, great messaging from county AIS staff, soil water, many people doing communications and messaging and doing important on the ground work. But I think our unique role is sort of having that connection to the research so that we're doing the research and then trying to translate it into these applications.
Yeah, and I think I'd add, I think another thing that we're doing is like we're a connection point across the state. So the local programs are important and it's really great that they're happening, but at the same time, like they can only work within their boundaries, right? So it gives a way to kind of create and build these connections. So I don't view extension or our programs as a replacement for any local effort. I would see it as hopefully a way to like enhance each other's efforts. And I think we do that. Like I said, like Starry Trek 2, like that's a partnership with those exact same organizations. We would not be able to have a statewide event like that. But the benefit for them is that we're able to provide the central coordination point. I think it takes a lot of work off their shoulders too, where they do have some work to do leading up to it, but I think not nearly as much without like the central coordination role that we end up playing. So I think that's part of that piece as well. And in terms of like, could we use more educators? I have an entire document with a list of other programs that I would love to start, but do not have the time to do. So yeah, totally, there is more work that could be done and like more needs that could be met with more people.
And then one last thing is we have a lot of autonomy and freedom and flexibility. And so we can recognize a need and pretty quickly spin up a program. Whereas if we were, working for a state agency, we wouldn't have that level of freedom.
All right, well, let's thank Dan and Megan. This has been terrific. Appreciate it.
Thank you.